Okay guys, so this chapter is chapter 8, and we're going to be covering the metabolic process called photosynthesis. So to start off with, this is a figure from your book showing two different things. The first thing is in the ocean. This is showing the amount of chlorophyll A. So before we get too deep into this, let me just tell you that chlorophyll A is going to be the main photosynthetic pigment molecule that's going to be responsible for absorbing light from the sun and therefore carrying out photosynthesis. So photosynthetic organisms like plants will have this molecule called chlorophyll A, again a pigment molecule that absorbs sunlight. And so this is showing the relative concentration of that pigment molecule found in aquatic ocean environments. And over here, this is showing the relative amount of land vegetation. And the darker green to maybe blue that we get on land means more vegetation. And as we go lighter into these brown shades, that's minimum land vegetation. And again, in the ocean, the dial kind of goes in the opposite direction. So this is the milligrams of chlorophyll per cubic meter. And so you can see that many aquatic environments are around here. And as we get towards land, you can see that the amount of aquatic photosynthetic material organisms, whether it's algae or plants or cyanobacteria, they're going to get more densely populated as we get towards land. And even some regions on land look like they might, very close to land, near these borders have some deep reds. That's kind of interesting. So you can see that photosynthesis is not just land plants. In aquatic environments, photosynthesis is happening as well. Again, as you get closer to land masses, the amount of photosynthetic activity goes up. Okay. So again, photosynthesis is going to be this process in which photosynthetic organisms convert the energy found in sunlight to the energy stored in organic molecules. So this is going to happen all over the globe, but again, the concentration of these types of organisms will be close to land or on land. So as an overview, again, photosynthesis, it's going to be a metabolic process, so it's a pathway, lots of intermediate steps where we're going to convert energy from the sun, which is in the form of wave energy, to the energy stored in the chemical bonds of food, right? Organic molecules like glucose. Okay, so we take energy from the sun and we convert it to chemical stored energy, or potential energy. All life on our planet depends on this process of photosynthesis. All organisms depend on photosynthetic organisms. We not only depend on them for energy, but these photosynthetic organisms are also able to fix carbon. So they're going to take carbon dioxide from the atmosphere and fix it, bring it into organic molecules. And we know that, organisms like us, we depend on not just energy, but also carbon for organic molecules that our bodies are made of. And so therefore, we depend on plants for kind of a multitude of things, right? We depend on them for oxygen, for food, and for carbon for our organic molecules. So in sum, down here, this summary statement, the energy of sunlight, it's going to be captured by photosynthetic organisms like plants, and it's going to be used, we're going to see, to energize electrons within these plants, and then that energy stored in those electrons will be converted into organic molecules. Okay, photosynthesis powers 99% of the Earth's ecosystems, so we heavily rely on this process. Okay, so who does photosynthesis? Well, we've got plants land plants like the one pictured here. This is a fern. This is actually what we're going to be looking at next week in lab, fern and moss, and other land plants like angiosperms, which are flowering plants, and gymnosperms, which are cone-bearing plants. We'll be talking about all of those in both lecture and lab in the weeks to come, in our final 2 weeks to come. So land plants definitely do photosynthesis. What else? Algae. Aquatic algae, which we can see here. All this green biofilm that we have here is all aquatic algae. And then lastly, if you remember back to this picture, this is right from chapter, I think, 1, I believe. This is a picture of cyanobacteria. So these are photosynthetic prokaryotic bacterial cells. All of these organisms, land plants, algae, cyanobacteria, are capable of performing photosynthesis. Okay, we would call these guys autotrophs. So autotroph means self-feeding, actually. So what that means is that they make their own food, and that term photo is they use sunlight, energy from the sun to drive that process. So a photoautotroph is going to use energy from the sun to drive the production of organic molecules. Okay. Heterotrophs are organisms like us, animals, fungi, most protists. We cannot make our own food. And so therefore, again, we rely on photosynthetic organisms like the ones pictured here. Now, sometimes students will say, yeah, so plants and algae and cyanobacteria, they do photosynthesis, but they don't do cellular respiration. That's not true. All organisms, including plants, algae, and photosynthetic cyanobacteria, do cellular respiration. That's common to all organisms. In fact, all cells of all organisms are capable of cellular respiration. Okay, so here is an overview of photosynthesis, and we've got pictured here, this picture here on the, let me just bring this down. On the right, we have a picture of a photosynthetic organism. Here is a tree, right, a land plant. Keep in mind, again, that photosynthesis is a multi-step process, so we call it a metabolic pathway. Even though we're going to write a very simplified equation for the process, there are a whole lot of steps that happen in between. Okay, a whole lot of intermediate molecules are produced before we get to our final end products. So, the beginning, when we look at this equation, we say that the substrates that are going to be, again, on the left-hand side of that arrow of the equation are going to include sunlight. So there's our energy source, carbon dioxide, and water. So let's quickly talk about where those things come from. So sunlight obviously is going to come from the sun. It's going to come down, it's going to hit our photosynthetic organism, and the green parts, and sometimes other colored parts as well, but the parts of the plant which contain chlorophyll. Okay, and so chlorophyll A is a green photosynthetic pigment molecule, and it's going to be responsible for collecting sunlight. And so in this tree pictured here, it is going to be the leaves, or in this case, these leaves possibly are modified. So the green structures, right? And so the photosynthesis will not be taking place in, for example, the branches or the stem or the roots of this particular plant, because there are no photosynthetic pigment molecules or cells capable of carrying out photosynthesis in those organs. So we see that leaves, when we look at plants like this, are going to be the major photosynthetic organism. And that's true for most plants. Leaves will be the organ of photosynthesis. And so leaves will absorb sunlight. And again, we'll talk about that specific molecule, chlorophyll, in just a minute. We'll look at it a little bit closer and talk about how exactly that does that. So what else is required? Carbon dioxide. So how does carbon dioxide get into the plant? It is going to get in again through leaves. We looked at leaves in a little bit of detail in lab. We are looking at leaves in detail this week in lab. And in this chapter, we will also look a little bit closer at the structure of a leaf, and then we will spend some time looking at leaves during the last 2 weeks of lecture as well. And so if we look at leaf structure, in the epidermis, there's going to be these cells called guard cells, which will open and close these structures called stomates. And so stomates are going to permit gas exchange. And so carbon dioxide is going to enter into a leaf through a stomate. And again, I'll show you that in a picture a little bit later. So that's how the carbon dioxide gets in. It gets actually right in through the leaf. Sunlight is absorbed by the leaf. How about water? Water is going to have to come in up through the roots. And so down here in the soil, we will have water as well as dissolved minerals. And those will be taken up into the roots. And the roots, after taking up that water and dissolved minerals, will then transport those substances, in this case up the plant, to the leaves, where photosynthesis will take place. The structure inside of the stem and the root that is going to be carrying that water, those are called xylem. Okay, so that's the actual tube-like cells that are responsible for transporting water up a plant. So xylem. Okay, so there's sunlight, carbon dioxide, and water. Now the products of photosynthesis, oxygen is going to be a waste product, and then the important product here, simple carbohydrates like glucose, which are going to be high energy storage molecules. Okay, we already learned about cellular respiration, so when we say what would those carbohydrates that are high in energy be used for? Well, they're going to be used in cellular respiration to produce ATP. And then we know what ATP does. It powers every cellular process that requires energy. Okay, so how does that oxygen leave? Well, that oxygen that is produced, it can be used in cellular respiration, but most of it gets let out. Okay, if oxygen is required later for cellular respiration, it'll actually come in through the roots. So the oxygen that's produced by photosynthesis is going to exit again through stomates. So the same way that carbon dioxide got in, oxygen's going to get out. Okay. Okay. So here is our very simplified equation for photosynthesis. Okay, so we've got carbon dioxide, we've got 6 of them, so 6 CO2 plus water, 6 H2O, plus sunlight. So energy from the sun is going to yield simple carbohydrates like glucose, C6H12O6, and oxygen, so 6O2. If you look closely, if you were to turn this arrow around and throw in ATP at the end, energy, right? It is the exact opposite of cellular respiration. Okay, so if you wrote this in the reverse and actually put the energy at the end here, again, it would be the equation for cellular respiration. So we're transforming energy. We're taking energy from the sun and we're capturing it and converting it into energy stored in the bonds of carbohydrates like glucose. Okay, so again, where does it occur? This process is going to occur in our plant organ called the leaf. Okay, so basic plant structure, plants are composed of roots, stems, and leaves. If it's a flowering plant, you also have flowers and fruits. If it's a cone-bearing plant, you have cones. So each organ is going to have a specific function. Most of the time, leaves are going to be specialized for photosynthesis. That is going to be their main role. If we take a look inside of a leaf, the middle layer of cells in between the epidermis, so sandwiched in between there, are going to be called the mesophyll parenchyma cells. And again, in lab this week, you'll take a nice peek at that. Inside of these parenchyma cells, these mesophyll cells in the middle, you are going to find chloroplasts. And so this is what a chloroplast looks like here in this image. So a chloroplast is going to be a double membrane structure. And again, if you remember back to chapter 4, we looked at this in a little bit of detail. So it's going to be composed of 2 membranes, both an inner and an outer membrane. There will be a space in here, which is pointed at right here, called the intermembrane space. And then on the inside, there's going to be a fluid surrounding these structures called the stroma. Okay, so it's a watery fluid or an aqueous fluid. And then what we have are these disc-like structures called thylakoids. And if we kind of chop this disc down the middle, we get a structure that looks like this. Each of these discs has an inner space called the lumen, the thylakoid lumen. Okay? If we have a whole stack of thylakoids, these are all interconnected, and the stack is called a granum. Okay, this is going to be the structure inside of these mesophyll parenchyma cells inside the leaf that is really going to be responsible for photosynthesis. So both phases, which we're going to hear referred to as the light reactions and the light independent reactions, are going to occur inside of the chloroplast. Okay, so these structures are going to become really, really important. We're going to see that in the thylakoid membrane, right, surrounding the lumen, so the green part here, the red in the middle is going to be the lumen, and the green on the outside is going to be the membrane. It's going to be in that structure, the thylakoid membrane, where we're going to find that pigment molecule called chlorophyll, and we are going to find proteins that are part of, look, another electron transport chain. So lots of similarities here, structurally and function, between cellular respiration and photosynthesis. We're going to find the same kinds of things have evolved for both processes. It's pretty neat. Okay, so primary organ of photosynthesis is the leaf. The primary organelle where this is happening is inside of the chloroplast. And again, I wanted to just reiterate what I said in the last slide. How does carbon dioxide get in? How does oxygen, the waste product, get out? Those gases will move through stoma or stomata. Those are going to be openings on the epidermal surface of leaves. Okay, so photosynthesis can be broken down into two main parts or two main phases. The first phase is called the light dependent reactions, and the second phase, it used to be called the dark reactions or the dark phase, but that was a little bit misleading because it makes it sound like it requires darkness in order to occur, and that's not true. So we call them either the light independent reactions, or we can refer to them as the Calvin cycle. Okay, so the light dependent reactions happen first, and the Calvin cycle really happens second. So the light dependent reactions, as the name implies, require light. And so energy from the sun, okay, is going to be absorbed by the leaves. Specifically, we're going to be inside of this structure here, which is the chloroplast, inside of leaf mesophyll parenchyma cells. And so if we look here at the stack of thylakoids, the granum, that light is going to be absorbed by chlorophyll, which will be found inside of the membranes of these structures, so the thylakoid membranes. This process will also require water. So we've got light and water, and oxygen will be a byproduct. But more importantly, the other products from this first step, the light dependent reactions, are going to be ATP and NADPH. NADPH is NADH and FADH2's cousin. Okay, so if you remember what NADH did, right, it was an electron shuttling molecule. That is the same job of NADPH. It's also going to carry high energy electrons from one area to another. And you know what ATP is, right? So we just quickly converted the energy from light wave energy into short-term energy storage molecules, ATP and NADPH. That's what's going to happen in the light-dependent reactions. Again, these are taking place inside of the thylakoid membrane, inside of chloroplasts, in the mesophyll parenchyma cells, in the leaf. That ATP and NADPH is then going to drive, those will be the reactant molecules that are going to drive this next step, along with carbon dioxide. This is going to happen in the stroma, so that aqueous fluid surrounding the granum, again, still inside of the chloroplast, and still inside those leaf mesophyll cells. So the Calvin cycle, again, is also known as the light independent reactions. So again, the energy, ATP and NADPH, those are short energy storage molecules. That's going to drive this reaction in which we're going to take carbon dioxide and convert it into this GA3P. This stands for simple carbohydrate molecules. And this will then be converted into glucose. Okay. The products here are glucose, and we recycle ADP, inorganic phosphate, and NADP plus so that they're ready for the next round of the light-dependent reactions. Okay, so it's pretty simple to see in this figure. We're going to go through the steps of both the light-dependent reactions and the Calvin cycle in a little bit more detail. So those are our two parts or two phases. So right here, I want you to take a break, and I want you to take out your Are You With Me, and you can answer the first Are You With Me on that in-class assessment. When you're finished, you can continue.
Okay, so like I've told you in past videos, some of these videos were made last semester, and so were a little bit off of timing. So I think I mentioned during the beginning of this that we were looking at leaf slides this week, plant tissue slides this week, and we're actually not going to be looking at them for another week or so, where we start to focus on plant tissues and plant structure and function during the last couple of weeks here of the semester. So you haven't looked at leaves yet. However, you can go ahead and you can find a picture of a leaf right in your lecture textbook, so right in chapter 30, I believe. There's a couple of pictures of leaves. And so again, photosynthesis takes place in leaves. Those are the main organ of photosynthesis in most plants. And in the mesophyll, which are the cells in between the lower and upper epidermis, that's where photosynthesis will be taking place, right inside of those cells. They're called mesophyll or parenchyma cells. Okay, so again, in this third question here where I say make a labeled sketch of a leaf, go ahead and take a look at chapter 30 and try your best to make a nice labeled sketch of a leaf. Okay, and again, you're going to label those cells that are in the mesophyll. And if you draw a dicot leaf, you haven't heard that term yet, but you will find that dicot leaves have both a spongy as well as a palisade parenchyma layer. Okay, so both the spongy and the palisade cells that you see are collectively called the mesophyll cells. Okay, if you're looking at a monocot leaf, which is a different type of leaf, there is no distinction between palisade and spongy. It's just all mesophyll. Okay, so again, even though we haven't looked at leaves yet, go ahead and find a picture of a leaf, make a sketch, and make sure you label those mesophyll cells. And remember, again, this should be your own sketch, so don't just pull a picture off the internet and label stuff. You want to actually sketch it yourself. Okay, you don't have to be extremely artistic. I'm not looking for artistic ability here. I'm just looking for, make sure that structures are there. Okay, so go ahead and take a look. I believe it's chapter 30 of your textbook that covers organs, which leaf is the photosynthetic organ. All right, before you go on too, I have a couple of questions for you. So here's the first one, the first slide. Okay, so here we go. In which organelle does photosynthesis take place? So not which organ, not which cell type, but which organelle. Okay, so which organelle is it? Is it the mitochondria? Is it the chloroplast? The endoplasmic reticulum? Or does photosynthesis take place in the cytoplasm? Go ahead and answer that. Okay, if you need more time, remember, just pause me. So the organelle in which photosynthesis takes place in is called the chloroplast. And inside the chloroplast, different parts of photosynthesis are going to take place in different structures. Okay, so remember, the organ of photosynthesis is the leaf, usually. There are exceptions to that rule. And inside the leaf, the cell type, the cells are going to be mesophyll cells. Okay, parenchyma, mesophyll cells, either one. And inside of those mesophyll cells, the organelle of photosynthesis is called the chloroplast. Okay. Remember, this is inside of plants. If we're talking about cyanobacteria, those are prokaryotes. They actually lack organelles, so we're going to be talking about totally different structures. But inside of eukaryotic plants, this is where photosynthesis takes place. Next question. Which products result from photosynthesis? Are the end products oxygen and carbon dioxide, chlorophyll and oxygen, sugars or carbohydrates and oxygen, sugars or carbohydrates and carbon dioxide? So what are the products of photosynthesis? Think about that for just a minute. And hopefully you answered green. Yep, so glucose, which is a sugar or carbohydrate, it's a monosaccharide, is definitely a product of photosynthesis, right? That's a long-term energy storage molecule. That's the most important end product of photosynthesis. This is how plants are going to store energy, okay? And then oxygen. Yes, oxygen is a byproduct of photosynthesis. Remember that water and carbon dioxide, water is going to be the reactant molecule, as well as carbon dioxide and light, and those are going to in turn produce sugars, carbohydrates, and oxygen. Okay, we'll actually see in the next couple of sections where exactly that oxygen is produced. Okay, next question. Which organism doesn't perform photosynthesis? So we've got fungi, cyanobacteria, plants, and algae. Which one of those does not perform photosynthesis? So hopefully you answered fungi. Fungi are heterotrophic. They do not make their own food. The rest on here, so cyanobacteria, these are going to be photosynthetic bacteria, as well as plants, as well as algae. Okay, they're going to all be photosynthetic. Fungi are not. Next question. Which is the main organ of photosynthesis in most plants? Is the main organ going to be stems, leaves, roots, or flowers? That was an easy one, right? Hopefully you answered leaves, okay? Now again, it says most plants. We will see in later chapters as we talk about plant structures and functions that not all plants have photosynthesis taking place in their leaves, especially if they have modified leaves or modified stems, okay? But for the most part, photosynthesis is going to take place inside of leaves. So we call leaves the organ of photosynthesis. Okay, that's the last one. So on to the next section, and then I'll be back with some more questions for you.
Okay, so when we're talking about the light-dependent reactions, again, one of the reactant molecules required to drive this process is energy from the sun. So let's take a quick look at what that energy looks like. So the sun is going to emit what we call solar energy, or electromagnetic radiation, in the form of waves. And so wave energy looks kind of like this. So you've got these waves. The top of a wave is called the crest, and the bottom of the wave is called the trough, and the distance between two consecutive crests is called the wavelength. Okay, and the wavelength tells us how much energy that is possessed. The shorter the wavelength, the higher the energy. The more spread out those two crests are, so the longer the wavelength, the lower the energy. And so when we look at wave energy coming from the sun, we are going to have some of that energy within what we call the visible light spectrum. And so if I take this part right here, the visible light spectrum, and I kind of spread it out, you can see, you're probably familiar with this, Roy G. Biv, right? This is visible light. So red, orange, yellow, green, blue, and violet. And we know that there are certain regions within the visible light spectrum that are going to be wavelengths of light that are going to be absorbed by photosynthetic organisms. So that pigment molecule, chlorophyll, and that's not the only pigment molecule, we'll also talk about carotenoids. We know what type of light they will absorb. So in this figure, it shows the different lights as part of the visible light spectrum, and it also shows their wavelength. And so the violet is going to have a shorter wavelength. So if you look at the distance between two consecutive crests, they're pretty close together. And if we go out here to red, we find longer wavelengths, so a longer distance. And so if we were talking about energy, the shorter the wavelength, the more energy that type of light will possess. As we go up here towards red, or if we look at our visible light spectrum, we go down here towards red, that type of light is going to possess less energy. Okay, so let's look at our pigment molecules and where they absorb. So there's 3 pigment molecules. 2 of them are chlorophylls. So we have chlorophyll A and chlorophyll B, shown here, chlorophyll A and chlorophyll B. And this last one is called beta carotene. It's a carotenoid. And so chlorophyll A is really the main photosynthetic pigment molecule, but there are also chlorophyll B and carotenoids that are part of, that are found in the thylakoid membrane that are part of these photosynthetic systems. So they're important as well. And so again, all 3 of these photosynthetic pigment molecules are responsible for absorbing light. They're going to, each of them, absorb specific wavelengths of light within that absorption spectrum. We've measured this using an instrument called a spectrophotometer. We can actually put these photosynthetic pigments into this machine, and we can hit it with the whole visible light spectrum, and we can see which ones are absorbed and which ones pass through, and we can then say which ones the particular pigment molecule is going to absorb and use to drive this process. So again, photosynthetic organisms like plants, they're going to have a mixture of these photosynthetic pigments. So let's take a look at each of them. So if we look here, this figure showing the absorption spectrum, again, this was gathered using one of those machines called a spectrophotometer. We've got chlorophyll A as our solid white line, chlorophyll B is the larger dotted line, and then beta carotene, which again is a carotenoid, is going to be the small dashes. And what we find is that chlorophyll A is going to absorb in the blue-green spectrum, so around, between 400 and 450, really peaking up here, towards maybe 425, 430. And it's also going to absorb down here in the orange-red part of the spectrum, okay, which is closer to 650 to 700 nanometers in wavelength. So it absorbs at 2 ends. And then we've got beta chlorophyll, again, that heavier dashed line, which is going to absorb here, again, in the green part of the spectrum, around 450, 460 is where it really peaks. And again, it will also absorb here around 650. So both chlorophyll A and B will absorb in the green to blue region, as well as the orange-red region of the visible light spectrum. Beta carotenoid is going to absorb in the green-yellow spectrum, so it's going to peak around 500. So again, there's this mixture of pigment molecules all there to try to get as much energy out of that sunlight as possible. Okay, so let's look at the light-dependent reactions. So again, what's going to happen here is these pigment molecules are going to absorb energy from the sun, right? And it's going to convert that energy into the chemical energy in the form of NADPH and ATP. And we know that both of these are going to be short-term energy storage molecules that are going to be used in the second step of this process, the Calvin cycle. Okay, so how exactly does this happen? So again, we're inside of a leaf, which is our main photosynthetic organ. Inside of the leaf, we're going to be in the middle, the mesophyll, in the parenchyma cells. And inside those cells, we're looking inside at chloroplasts. Okay? Inside of these chloroplasts, there will be thylakoids. And in the thylakoid membrane, there are going to be proteins involved in this process of absorbing light and then converting that energy into ATP and NADPH. Those proteins are going to be really multi-protein complexes that are going to be associated with chlorophyll A, chlorophyll B, and carotenoids, so those pigment molecules. There are actually two types of these multi-protein complex systems found in the thylakoid membranes, and they're very nicely named photosystem 1 and photosystem 2. Now, the 1 and 2 refers to when they were discovered. So photosystem 1 was discovered first, and photosystem 2 was discovered second. But they actually work in the reverse order. So photosystem 2 is going to really work first, and then photosystem 1 really comes after it. So that's a little bit confusing. So we'll take a look at that. Okay. So again, even though photosystem 1 was discovered first, it actually acts second. So in this figure, what we've got going on here is we've got the first part of the reaction, which happens in figure A. This shows photosystem 2. If you look here, you've got the thylakoid membrane, right? The stroma is on the outside. This is the fluid that surrounds the thylakoid structures. And then we've got the inside called the thylakoid lumen. Okay, remember all of this is inside of a chloroplast. This green structure with the blue inside of here, this whole thing is photosystem 2. And in part B, this whole thing is photosystem 1. Okay, so again, photosystem 2 is going to work first. So what do we have here? We have this big protein hanging out in the thylakoid membrane, and you see all these little green circles here. Those represent pigment molecules. So you got your chlorophyll A, your chlorophyll B, and your carotenoids. They're all associated with this protein, and their job is to absorb light. Okay, so what's going to happen? Light's going to come. That light will then be absorbed by these pigment molecules. That energy gets passed from one pigment molecule to another. Okay. Eventually, that energy gets passed to the center of the photosystem. And in the very center of the photosystem, there's going to be it's a doublet of pigment molecules. These will be chlorophyll A molecules. And when that energy gets passed here, electrons in these pigment molecules, they get excited. They kind of jump out to outer energy levels. And instead of dropping back down, which would happen if they then pass their energy to the next pigment molecule, they don't do that. Instead, when this electron jumps up and it gets excited, this right here, called the primary electron acceptor, it grabs onto those high energy electrons. And so that energy doesn't get passed to another pigment molecule. Instead, it gets passed here to this primary electron acceptor, not a pigment molecule. Okay? We'll talk about what happens to that energy in just a second. But before we do that, now this doublet, these pigment molecules lost electrons. Well, that's not good. Those electrons need to be replaced, and that's where water comes into play. And so here we have water, which, remember, water and light are our two reactant molecules that drive the light-dependent reactions. And so what happens is this water molecule is going to be it's going to be lysed, it's going to be split, and it's going to give electrons to these pigment molecules that just lost their electrons. So the role of water in photosynthesis is to replace electrons in photosystem 2. Okay, the product here would be oxygen and then some protons. Okay, so there's our waste product of oxygen. Oxygen is produced in that very first step. Okay, let's see what I missed. So again, pigment molecules in photosystem 2 are going to absorb light, pass that energy to the very center of the photosystem 2 complex, where there's going to be, again, a pair of chlorophyll A pigment molecules that are just waiting for that energy. Those then undergo oxidation upon excitation. So electrons are going to jump up and be stolen, be taken from those chlorophyll A molecules by that primary electron acceptor. So they give up an electron. And we know that electrons represent energy. So what just happened is light was captured and energy was transferred from the pigment molecule to the primary electron acceptor. So now that primary electron acceptor has that energy. Okay. I think we'll hold off on looking at photosystem 1 in detail for just a second. Just to show you, it's also in the thylakoid membrane. This would be the stroma on the outside, and this would be the thylakoid lumen on the inside. And photosystem 1, just like photosystem 2, is a protein complex that contains pigment molecules. Now, if you notice the number next to the photosystems, so this is photosystem 2, P680, and this is P700, that's showing kind of the average light wavelength in nanometers that those chlorophyll, or those pigment molecules absorb best at. So they're slightly different there. The function is pretty much the same. So again, without going through it in too much detail, you can still see light comes in, it gets captured by pigment molecules. That energy gets passed to the center where we have a pair of chlorophyll A. Electrons get excited and they are going to be captured by the primary electron acceptor. So it's really the same thing happening in both photosystems. In photosystem 2, the electrons are replaced by water, but in photosystem 1, the electrons are not replaced by water. They're replaced by something else. So we'll talk about that in just a second. Okay, so both really have the same structure. They're proteins found in the thylakoid membrane that have associated pigment molecules. Those pigment molecules absorb light from the sun. That energy is passed to chlorophyll A, a doublet in the center. So two chlorophyll A molecules found in the center of that protein complex. And electrons from that chlorophyll doublet are going to become excited and captured by the primary electron acceptor. And of course, those electrons have to be replaced. Photosystem 2, they're replaced by water. Okay. Shift that a little bit. Okay. So in this figure, we're showing inside of a chloroplast, we've got the stroma, the liquid all around, and this phospholipid bilayer represents the thylakoid membrane. On the inside, we've got the thylakoid lumen. Here is photosystem 2. Here is photosystem 1. So again, both of them reside within the thylakoid membrane. Let's look at photosystem 2 first. This one acts first. Okay. Here's light being shown here by this little lightning bolt-like thing. Light from the sun comes in. We know that it gets absorbed by our pigment molecules, those little green circles. That energy gets passed to this doublet, these two chlorophyll A molecules found in the center of this reaction center. Electrons get excited and get captured by this little molecule up here called the primary electron acceptor. Electrons are replaced by water. Okay, we've just read all of that on the previous slide. Here's what we didn't see. So those electrons now, up here, that were captured by the primary electron acceptor. Let me get my pen, see if I can do this. Oop. Nope. Okay. So right up here, we have our primary electron acceptor. This captured those high energy electrons from chlorophyll A, which was down here. Okay, so now what happens to those electrons? They're going to be passed here. This is called plastiquinone. Don't worry about it. These are parts of the electron transport chain. So this PQ, this cytochrome, and this PC down here, these are all just parts of the electron transport chain. Don't worry about their names. What do they do? So we just had electrons that were going to be given from this primary electron acceptor to the first protein here in our electron transport chain. So we say that this just got oxidized while this got reduced, right? So we had a redox reaction just happen, a passing of electrons from one molecule to another. Okay, so the energy of those electrons was passed from here to here. So now that energy, a little bit of it, is going to be captured by this first PQ, this first protein complex of the electron transport chain. And that energy, it's going to be used, guess what, to pump protons, the same thing that we saw in cellular respiration. So those protons will be pumped across the thylakoid membrane from the stroma into the thylakoid lumen. Okay? Next, those electrons are passed to the next part of the electron transport chain, right here. CYT, it stands for cytochrome. Again, a little bit of the energy of those electrons will be lost, captured by this cytochrome complex to pump protons from the stroma into the thylakoid lumen. And then those electrons get finally passed down here to the last part of this electron transport chain. And then finally, those electrons are going to be passed to complex 1 to replace the electrons that were lost. Okay, so the energy from the sun was captured by complex 2 and used to drive this electron transport chain, which we've now built up a proton gradient. Look at all the protons now that we have here in the thylakoid lumen. And if we look out here in the stroma, there's really none. And so we just took energy from the sun and we used it to build up proton motive force. What happens next? Well, let's skip this for a second and let's come down here. Does this look familiar? This again is called an ATP synthase. It's the same one that you saw in cellular respiration. It's going to be how protons move down their gradient from high to low concentration. There's no way that these positively charged protons could move through this lipid bilayer on their own. So they need to move through the ATP synthase, and when they do, the ATP synthase is going to crank out ATP. So remember that that's one of our products, one of our products from the light-dependent reactions is ATP, and it's made by the ATP synthase, and it's already going to be in the stroma, where it's going to be required to drive the Calvin cycle. So we say that photosystem 2 is responsible for the production of ATP by first building a proton gradient. Okay, so let's go to photosystem 1. So photosystem 1 we know gets energy from the sun, right? Light energy is absorbed by those pigment molecules. That energy is passed to chlorophyll A right here in the center. That energy is going to be captured in the form of electrons up here by the primary electron acceptor. So the same idea. The electrons here that are lost, they do not get replaced by water. Instead, the electrons that have traveled down this electron transport system or chain are going to be passed from this final protein to chlorophyll. So that's how those electrons are replaced. We've got water here for photosystem 2. That's going to be replacing electrons. And for photosystem 1, those electrons get replaced just by the electrons that were lost from photosystem 2. Okay, so now these high energy electrons, they get passed down another little electron transport chain. But this electron transport chain does not build up a proton gradient. Instead, it's just going to pass those high energy electrons to NADP, forming NADPH. Okay, so photosystem 2 produces a proton gradient, which leads to the production of ATP, where photosystem 1 is going to lead to the production of NADPH. Both photosystems are going to get their energy from the sun. That's it. So both ATP and NADPH that are produced here, notice how they're both produced in the stroma. So they're already in the right spot, the right compartment, the right place for the next phase of photosynthesis, which we know are the light-independent reactions, which means that they can happen in the presence of sunlight or the absence, or they are also known as the Calvin cycle. that kind of fast on that one slide. So this next slide, again, just has the words for you if you want to read them again. Okay, so again, photosystem 2 produces ATP, where photosystem 1 is responsible for the production of NADPH. Both photosystems will use light energy and convert that light energy into either ATP or NADPH. Remember, those represent energy storage molecules, short term, right? Your cell really can't build up NADPH and ATP and have those molecules be long-term energy storage molecules. And so instead, that's why we have to take that energy and put it into organic carbohydrates like glucose, because those are long-term energy storage molecules. ATP and NADPH are not. Okay, so here is that second. Are you with me? So again, you're going to want to fill that out, and as soon as you're finished, you can go on.
Okay, so here's some more questions that we're going to answer together. So after you've finished those Are You With Me, you can go on to the next slide. And so will I. Okay, so just a couple questions for you. In which part of the chloroplast are photosystems 2 and 1 found? Notice how I put 2 before 1. That's because that's the order in which they really work. Okay, so where do we find them? Do we find them in the thylakoid membrane? Or do we find them in the stroma? Are they located in the inner membrane of the chloroplast? Or are they located in the thylakoid lumen? So where do we find photosystems 2 and 1? Again, both of those are part of the light-dependent reactions. They're going to be found in the thylakoid membrane. Remember, they are going to be kind of loaded with pigment molecules that are going to be responsible for absorbing light energy from the sun. Those pigment molecules become excited. They end up passing their high energy electrons to the electron acceptor molecule, and then those electrons get passed again down the electron transport chain, either generating a proton gradient, which makes ATP, or generating NADPH directly. Okay, so remember, those photosystems are extremely important. They capture light energy and generate the chemical stored potential energy, NADPH and ATP, which will then be used in the second part, which we haven't talked about yet in too much detail. The second part of photosynthesis is either going to be called the Calvin cycle or the light-independent reactions. Okay, the energy to drive that second step is produced here. Okay, and again, photosystems 2 and 1 are crucial components to that, and they're going to be located in the thylakoid membrane. So we're in a leaf, right, in a mesophyll cell, inside of a chloroplast, inside of that thylakoid membrane. That's where we're going to find photosystems 2 and 1. Okay, next question. What is the initial source of electrons for the chloroplast electron transport chain? Okay, so remember that electrons are going to be first lost by photosystem 2. They're going to be transferred to photosystem 1. And then photosystem 1 is going to take those electrons, and those electrons will be passed to NADP, forming NADPH. And so with every round of the light-dependent reactions, electrons are lost in the formation of NADPH. Okay, and they're lost from photosystem 2. Okay, so how do those electrons get replaced? What is the source of those electrons? Where do they come from? Do we get those electrons from water, from oxygen, from carbon dioxide, or from NADPH? So go ahead and take a second to think about that. So hopefully you answered water. Remember that water, photolysis, right? Water is going to be split, and electrons are going to be given to the chlorophyll molecule, which gave up its high energy electrons. So they're always going to be replaced, replenished by water. That's the role of water in photosynthesis, to replace electrons that have been lost by pigment molecules of photosystem 2. Okay, next question. what is the direct role of pigment molecules? So remember, our pigment molecules are chlorophyll, A and B, and then carotenoids like beta-carotene. Okay, those are going to be involved here. What is their role? Are pigment molecules responsible for absorbing light, for generating NADPH, converting ADP and inorganic phosphate to ATP, or are pigment molecules responsible for carbon fixation? So what is the direct role of pigment molecules? Take a second to think about that. So remember that pigment molecules are directly responsible for absorbing light. When they absorb light, they're absorbing energy, okay, light energy or wave energy. That's their direct role. Yes, NADPH and ATP are produced as a result of that light absorption, but they're not directly responsible for that. They're only directly responsible for absorbing light. Okay? Remember, our pigment molecules generally are going to, the ones that we're looking at, are going to absorb in the blue-green region, slightly in the green region, and the red-orange region. Okay? And actually, most of that green light in the visible light spectrum gets reflected. And so that's why leaves usually appear green. Okay, that's the light that's really not being absorbed to a great extent, and that light is being reflected. Okay, next question. I think this is the last one. The end products of the light-dependent reaction include, okay, so what are the end products here of the light-dependent reactions? Water and carbon dioxide, ATP and NADPH, carbon dioxide and ATP, or oxygen and glucose. So not the overall products, but just the products here of the light-dependent reactions. So take a second to think about that. Okay, and so hopefully you answered ATP and NADPH, right? So these light-dependent reactions are going to take light energy, right, wave energy from the sun, and they're going to convert that energy into short-term energy storage molecules, ATP and NADPH, through a series of steps. These get produced. Okay, so there's that conversion, wave energy to ATP and NADPH, which is chemical stored potential energy. Those two products are then going to go on and drive the next stage, which is the Calvin cycle. Okay, so we have to first convert energy, use that energy then to make organic products, to make glucose. Okay, so on to that next part. So again, we've got one more section, I think, to go, and then we'll be back and I'll ask you some more questions. Okay.
Okay, so all of that ATP and NADPH that were made in the light-dependent reactions, those are going to represent, again, short-term energy storage molecules, not long-term, short-term. We need a long-term energy storage molecule. That's going to be the end product of this process. So all of that ATP and NADPH produced in that first set of reactions is going to be used to drive this second stage. Okay, which again is called either the light-independent reactions or the Calvin cycle, formerly known as the dark reactions. So for this part, again, the ATP and the NADPH are already in the right spot. They're in the stroma, and so that's where this Calvin cycle is going to take place. But the last molecule that's going to be needed, which is carbon dioxide, that is going to come in again through stomata or through stomates. It's going to enter into the mesophyll cells, and it's going to diffuse right into the stroma. So it's going to get right to the same place as that ATP and NADPH are located. And there are 3 stages. We'll talk about them briefly. The first is called fixation, carbon fixation, I should say, and then reduction and regeneration. Okay, so 3 steps here. And I'll tell you right now that all of the organic, carbohydrates, simple sugars, are produced by the end of the reduction phase. The regeneration phase is to regenerate the starting molecule. Okay, so here it is, and it's a simplified version. And so stage 1 we'll talk about first. This is the Calvin cycle. It's a cycle, just like the Krebs citric acid cycle was that we saw in cellular respiration. So again, it goes in the clockwise direction. We have our starting molecule here and here. And again, we're going to go through this cycle and we're going to regenerate our starting molecule. Okay, so it's a complete cycle, and each time it goes around, it'll be ready by the end for the next carbon dioxide molecules that are coming in. Okay, so initially we're going to start off with 3 molecules of carbon dioxide, which again we know come in through the stomata. And then we have 3 molecules of this, which again, you don't need to know structures or even really names here. So 3 molecules of what's called RUBP, okay? And then this enzyme right here, Rubisco, is a very, very, very important enzyme. It's probably the most prevalent enzyme on the planet. And it has a very, very important job. It is responsible for fixing carbon. So this protein, this enzyme, probably has the most important job of all the proteins or enzymes. It's responsible for taking carbon from the atmosphere and incorporating it into organic molecules, incorporating it into simple carbohydrates. So really, really important. Again, all of the carbon that we have incorporated into organic molecules comes from atmospheric carbon. And so this enzyme is really, really important. So the first thing that's going to happen is this Rubisco, this enzyme, is going to catalyze the reaction between carbon dioxide and RUBP. So again, that's the first stage here, carbon fixation. And at the end of that, we're going to have 6 molecules of 3-PGA. Okay? That's the first step. So fixing carbon. We just took carbon dioxide from the atmosphere and we incorporated it into these molecules. We ended up with 6 of 3-PGA. Again, you don't need to memorize names or structures. Maybe that enzyme though, Rubisco, you should know what that is. So the next step here is going to be reduction. So we've already fixed carbon in step 1. We've ended up with 6 molecules of this 3-PGA. Now we're going to take some of that energy that was produced during the light reactions. So we're going to burn 6 molecules of ATP, as well as some of that NADPH, again, also produced during the light-dependent reactions. And we're going to reduce the 6 molecules of 3-PGA to form 6 molecules of what are called GA3P. This is called glyceraldehyde 3-phosphate. 1 molecule of glyceraldehyde 3-phosphate will leave the Calvin cycle and will be used to make a half a molecule of glucose. So it takes 2 rounds of this cycle to produce 1 whole molecule of glucose. Okay, so there's our simple carbohydrates that are produced as a product. Okay, so by the end of the reduction phase, we've spent a lot of ATP and a lot of NADPH, but we've built a high energy storage molecule, right, representing chemical stored energy. The last thing left to do is to regenerate our starting molecule. And so the other 5 molecules of glyceraldehyde 3-phosphate, GA3P, are going to be used in this last stage, which again is called the regeneration of RUBP. We're going to need a little bit of energy here, 3 molecules of ATP, along with these 5 molecules of GA3P, so glyceraldehyde 3-phosphate, and we end up producing 3 molecules of RUBP, ribulose bisphosphate. Okay, so this will be available again for the next round of the Calvin cycle. So it takes 2 complete rounds of the Calvin cycle to produce 1 molecule of glucose. So it takes a lot of energy and a lot of reducing power. Okay, but in the end, we have our organics. We have our simple sugars, which are then going to be broken down in cellular respiration to yield that usable energy source, ATP, for our cells. Again, the reason why the ATP produced during the light dependent reactions isn't, just used to store energy is because it can't. It's not a long-term energy storage molecule, where glucose is. Okay, so here's a summary of the light-dependent and the light-independent reactions. Okay, all of them are taking place inside of leaves, inside of the leaf mesophyll cells, inside of that very specialized compartment called the chloroplast. Again, the light-dependent reactions are going to occur inside, really, the thylakoid membrane, although the thylakoid lumen is a very important space, right, for that proton motive force. And the Calvin cycle, or the light-independent reactions, are going to take place inside of the stroma. Now, once the sugar molecules are produced, they're either used in photosynthesis or they're stored somewhere in the plant. Fruits represent storage of carbohydrates. Cells can store carbohydrates as well. If we look inside of root vegetables, right, those are all storage compartments as well. So potatoes, sweet potatoes, beets, those are going to be storage molecules. Apples, those are where plants are going to be storing some of these sugars. We can also, again, open up the different structures inside of a plant and find starch storage structures in.
So my video got cut off there. You can find plant storage molecules inside of other cells within a plant as well. Okay, so we're going to go on to our last Are You With Me? So you're going to pause here and you're going to describe the steps in the light-independent reactions. Remember, a sketch always really helps as well. So a description and a sketch would be great. And then you're going to be finished with your Are You With Me's. You're going to go ahead and upload those to Blackboard. But before you do, I've got just a couple more questions for you. So after you're finished with this Are You With Me, come on back and we'll do those. Okay, so the first question here, we got 4. Which molecule must enter the Calvin cycle continually? So what has to be continually taken in by these plant cells and continually enter into chloroplasts in order for the light-independent, or the Calvin cycle, to continually take place? What's required? Okay, is it Rubisco? Is it RUBP, 3-PGA, or carbon dioxide, CO2? So hopefully you answered CO2. Okay, so the Rubisco is an enzyme that'll already be present, right? That RUBP is the first molecule that gets regenerated every round of the Calvin cycle. So those things don't have to be constantly taken in. And 3-PGA, that's an intermediate molecule, so we're not worried about that one. But carbon dioxide is a reactant molecule that the cells of the plant have to continually take in order for this process to continue. So CO2 is required. It's a reactant molecule for the Calvin cycle that has to be continually taken in by these plant cells. And how does it get in there? Make sure you know. CO2 is going to enter through a stomate. Okay, stomate, stomata, one's singular, one's plural. It's the same structure. So these are going to be pores in the leaf epidermis, and we're going to look at those in a little bit more detail when we get into chapter 30. But those are going to be pores that are going to allow for gas exchange. So CO2 will come in, and the oxygen, which is produced as a byproduct, is going to leave through that stomate as well. Okay, so CO2 is a required material. It's going to enter in through stomates and then enter into these mesophyll cells, get right to the chloroplast where it's going to be used during the Calvin cycle. It's going to be fixed, we say, during the Calvin cycle. Okay, so where in the chloroplast, we know that it happens in the chloroplast, but where exactly in there does the Calvin cycle take place? Is it going to take place in the thylakoid membrane, the thylakoid lumen, the chloroplast stroma, or inside of a granum? So take a minute there. Okay, so remember that the first stage in photosynthesis, so the light-dependent reactions, those took place in the thylakoid membrane. That's where photosystems 2 and 1 were located, and also the ATP synthase. And the thylakoid lumen was going to be that space where that proton gradient was built up. Okay, so those are important in the first stage. And a stack of thylakoids is called a granum. Okay, so none of those make sense here. The Calvin cycle, or the dark reaction, or the light-independent reactions, they're going to take place in the chloroplast stroma, right? That's the fluid that surrounds the granum, that surrounds the thylakoid. That's where the ATP and the NADPH will be, that were produced by those light-dependent reactions, and they're going to drive the Calvin cycle, which is taking place out there in the stroma. Okay, next question. Oh, let's move my camera so that you can really see these. Okay, what is the role of the ATP and the NADPH? What is the role of these two in the light-independent reactions? Where those are the products of the light-dependent reactions. What exactly are they doing in the light-independent reactions? Are they there to fix carbon? Do they have a direct role in carbon fixation? Are they there to supply energy? Are they there to regenerate RUBP? Or is their role to split water? So what is the direct role of ATP and NADPH? What are they there for? Give you a minute. Okay, so hopefully you answered to supply energy, right? These are short-term energy supply molecules, and they are there to supply the energy required in order for the Calvin cycle to take place, in order for carbon to be fixed. Okay, last question here, it's a long one. Which of the following statements is true? Okay, so in pink, in photosynthesis, oxygen, carbon dioxide, ATP, and NADPH are reactants. G3P and water are products. Okay, that's pink. Blue, in photosynthesis, chlorophyll, water, and carbon dioxide are all reactants. G3P and oxygen are products. Green, in photosynthesis, water, carbon dioxide, ATP, and NADPH are reactants. RUBP and oxygen are products. Or yellow, in photosynthesis, water and carbon dioxide are reactants. G3P and oxygen are products. So hopefully you answered yellow, right? So water and carbon dioxide are our reactant molecules along with light. And our products are going to be G3P, which stands for glyceraldehyde 3-phosphate, which goes on to make glucose molecules. Okay, you need a couple of rounds of that Calvin cycle to get a glucose molecule. And also oxygen are the two products. Oxygen's the byproduct, right? G3P is the organic molecule, which then goes on to make glucose. Okay, so that's it for this chapter. So again, make sure that you upload your Are You With Me's right to Blackboard and study. And then when you're ready, you can go ahead and you can take that quiz.